Welcome back, everybody. I don't know about you, but I really did enjoy that lunch. There was particularly the beef. That was my favorite part. Okay, we're ready to get started for another afternoon of information. Our next speaker is Dr. Agustin Lujan, a scientist in the fields of medicine, technology, and education. Dr. Lujan is currently working in Spain doing postdoctoral TANGO2 research at Professor Malhotra's lab in CRG Barcelona. Please welcome Dr. Agustin Lujan.
It's okay? Okay. So, as this is working a little bit weird. Well, my name is Agustin. I'm from Argentina. That's why I want to apologize to you about my English, because it's not as good as I would like to. So if you don't understand something, please stop me and say, Agustin, I'm not understanding this, and I will try to explain you a little bit better or in another way. So first of all, we have this disclosure that I don't have a conflict of interest, and this is just our point of view on the TANGO2 project. So today, the idea is to develop just two different things. One of them is to understand how a cell functions, because if you could understand that, you will understand all our results. And then we will move to what do we know about TANGO2 in our lab. This is moving alone. I'm not doing anything. You are moving the... okay, perfect, yeah. Was like an AI. I do this and you move it. It was really good. So we will start with the, to know how does a cell function. So to those that don't understand too much about biology, you can think that the cell, any cell in any organism, is the smallest way that we know, the smallest form that we know of life. So it's totally independent. And as every cell in our organism, we can think a cell as a small city. So a small city has, I have a lot of ticks to move. If you want, I can move the hand. Okay, good. So a cell is like a small city. You have boundaries, and those limits in a cell are made by something that we call plasma membrane. But it's like the limits of a city. So then, now we have our city here, is our cell. And now I will start to present you different organelles and different components of the cell to understand how a cell functions. So our first organelle here is called nucleus, and the nucleus is not more like, it's like in the real life are the libraries. So in the libraries we have all the information that we know as human beings and are packed in different books, and in the cells we have more or less the same. We have a nucleus with all the information that we need to produce all our cells, but packed in something that we call DNA instead of books. So next, as you can see here, this is the real life. And this is our nucleus in the cell. So then we have something that we call endoplasmic reticulum, and as I'm lazy, I will say ER, just that. And the ER is represent in real life all the professions that has the capacity to read the information that is in the library and to produce something. So in the case of the cells, we read the information that is in the DNA, and the ER has the capacity to produce something that we call proteins. So in real life, the ER could be the engineers, the architects, the builders, the chef, all the profession that has this capacity to read information and produce something. But then we have another very peculiar organelle that we call Golgi apparatus. And the Golgi apparatus is like the Amazon of the cell. So has the capacity to take this protein that we produce in the ER and then pack it and deliver it to the correct place of the cell that this protein is needed, even in the same cells or also secreting this protein outside to other cells. I never put Amazon there because they don't pay me, but if someone is working on Amazon, we can talk about it. So we have the Golgi apparatus that has this capacity to pack the proteins and send it to the correct place. So all this that I show you in this moment is called the conventional secretory pathway. It's the pathway that all the cells use to produce something that we call proteins and send it to the correct place. But every machine that we know needs something very specific to function, and it's energy. So in the cell, we have something that we call mitochondria, but the mitochondria in real life could be the power station of a city. So as you can see here, the mitochondria is like that. It's the power station. We produce all our energy, or most of our energy, there. And how we call it the energy in life, in biology, we call it ATP. So there are many different kinds of energy, but to be more specific, we will talk today just about ATP. So ATP and energy could be the same in the next slides. So we have many mitochondria, and mitochondria sometimes are making like a net, or sometimes are disaggregated in the cell, and are in different situations that both situations happen, are very dynamic and are all the time doing this fission and fusion. But then as animals, we have something very peculiar, and it's that we have the capacity to store energy. So we have an organelle that is called lipid droplet, that is like the batteries that we have and we use to, or not, in our phones and in our laptops, wherever. So lipid droplets are no more than, like the word says, a droplet, a ball, that is packed with lipids inside. And these lipids are like the batteries of the cell. So we can use energy in form of lipids that is inside these small balls. So then, you will kill me today, I think so. Next one, please. So how do we produce energy in cells? We have three main sources to produce energy in our cells. The first one are sugars. The second one are lipids. And the third one are proteins. So most of the cells prefer to use sugars to produce ATP because it's the fastest and more efficient way to do it. But then, if we increase the demand and the requirements of ATP in a cell, we will start to use other sources as lipids and proteins. So in the first case, we use sugars. We eat like 5 minutes ago. So the sugars that are in my blood goes to my cells, and in my cells are converted in ATP. So now, to speak, to move my hands, to watch you, I'm using a lot of ATP in my cells, and this ATP came from the sugar that I just ate. But if I start to running, for example, now for the next 2 hours, I will consume all the sugars that is in my blood and most that is in my cells, and I will start to use as a source to produce energy, lipids. And at the end, we will start to use proteins as the source of energy. It's not like black and white, but it's like we prefer to use sugars. We are always using some lipids for some pathways and also proteins, but then we move to drive more with lipids and then move to drive more by proteins. Yes. Yeah. So to produce ATP from sugars is something very easy for the cell. So we take the sugar from the outside and we make 2 different steps. One is outside the power station, the other is inside the power station to produce ATP. But in case of lipids, we need to activate them to move it from the lipid droplets to the mitochondria. So to move from the batteries to the power station, we need to activate those lipids to be used to produce ATP. How we do that? So we move a free fatty acid. When I say fatty acid, it's the same as I say lipid, or if the same as I say acyl chain. So we move a free fatty acid from the lipid droplets. We add a coenzyme A, that is a molecule that activates these lipids. And now this lipid, So as you can see in some presentations before, the coenzyme A comes from the vitamin B5, and after we activate this lipid has the capacity to enter to the power station to produce ATP. So now you know, all of you knows how a cell functions in your body. You know all the organelles, all the structures, and all the functions of each structure. And why I want to present you this, because if you can understand the function of any of these organelles, then you can start to think, okay, where is the protein that I'm looking for? If it is in the power station, is this in Amazon? Where is located? And if it is located in one of these organelles, maybe the function is related with this kind of functions that happens in those organelles. So then I want to present you what we know about TANGO2. So when I joined the lab of Professor Malhotra in 2021, he was interested to start watching what happened with the other TANGO proteins that he discovered in 2006, because for the last 15 years he was working on TANGO1, and it's a totally different protein, different gene, controls the secretion of collagen, and there is nothing similar to TANGO2. So what I did is what any of us could do today if we have a question, put it in Google. So I put in Google TANGO2, please, one before. So I put TANGO2 in Google, and I found this. I found a foundation that was working in a rare disease related with TANGO2, what was impressed. And the second, the second thing was a news in an Argentina newspaper talking about the only TANGO2 patient that we had in Argentina that also hooked my attention, and I said, okay, I want to be here, and I want to work on this topic. So after that, I started to read all the papers that were published until 2021, that were more 28 or 30. Yes, go. And so I learned a lot about the clinical features of TANGO2, as you can see here. Go 1, 2, and 3. But what caught more my attention was the apparition of the metabolic crisis. Because then, reading more about what was published, I realized that most of the metabolic crisis appears after situation that the cells feel stress. So under fasting, under infection, under high temperature, cells have something in common there. And there are two things that for me were in common there. One of them was that cells need to produce more ATP when we are under infections, under fasting, or when we have high fever, but also we produce more ROS. And I will explain you what is ROS, because ROS means reactive oxygen species. And as I told you, I'm lazy, so I will say ROS when I'm speaking about reactive oxygen species. So in both situations, the cells need more energy and is producing more ROS. But also was very interesting to see papers that show that when you add vitamin B, you stop the apparition of this metabolic crisis. And most of, or many of the B vitamins works at the level of lipid metabolism, carbohydrate metabolism, and protein metabolism. And some of them has also the capacity to scavenger the ROS production. So the first thing that we did was to, okay, if we want to work with this protein, what do we have to do? Well, let's see where it is in the cell. So we use a model of cells, of human-derived cells from different organs. In this case, all the work that I will show you are made in HepG2 cells, that are cells from the liver. And the first thing that I did was to put a flag in my protein and see by microscopy where is stuck this protein, where is located in the cell. So we did that, and also I put a marker in the mitochondria and in many other organelles, but I will show you, because we don't have too much time, all the positive results here. And as you can see here, in red I have the pointer. Now I can do something. So, as you can see here, in red is TANGO2 isoform 1. And I say TANGO2 isoform 1 because in human, TANGO2 has 6 different isoforms. That means that the same gene, the same information that we have in the DNA can produce 6 different proteins that came from the same gene. I really don't know, and I think that no one knows, sorry, if all the isoforms has the same function or not. So all my presentation is about isoform 1 of TANGO2. And as you can see here, I use here a marker for the mitochondria, for the power station. So in the type of experiments that I did, that this is confocal microscopy, when you have 2 proteins in the same place at the same time, for example, a green and a red, then when you make a merge, you see yellow, because both proteins are in the same place at the same times. That means that my protein of interest, that was TANGO2, was located in the mitochondria. I also found a sample that was in the cytoplasm of the cell, so was outside the organelles, or was not interacting with the organelles. So then we start to check what happened with the protein of interest that was TANGO2 in relation with other organelles in the cell. And what we found was that was in close conformation or interaction with other organelles like the ER that is here and the lipid droplets. So the lipid droplets here are in green and TANGO2 in red. And in this case, in green is the ER and in red is TANGO2. So always when you see something that is yellow, it's because you have both proteins in the same place and at the same time. But also here you can say, okay, but you are looking just in one plane. So you are looking in X, Y yourself. Well, in this case, we make different analysis because we have the capacity to make different cuts, the different slices, sorry, in the microscope, so we can make a 3D reconstruction of all the structures, and we can see if they are attaching one with another or not. So our first conclusion was that TANGO2 was predominantly attached to the mitochondria. There is also a pool in the cytoplasm, but what was interesting was that there was a transcendental interaction between the batteries, the builders, and the power station in our cell. Next one, please. So then, something very easy to do is to add a marker of our lipid droplets. So in the first slide, I show you that the lipid droplet is like a ball. So you have a monolayer of lipids, that those lipids are called phospholipids. And in the core, in the inside, you have another kind of lipids that are called neutral lipids. And you know these neutral lipids because if you make a test in your blood, you will see cholesterol and triacylglycerides. Those lipids are in the core of the lipid droplets, are the lipids that are inside the lipid droplets. So I add a marker to label these lipid droplets. And as you can see here, now the lipid droplets are all in red. But then I did something that is very common to do in science, that is to remove your protein of interest in the system. And then you see what happened with the cell. So if your cell has a function and you remove it from the cell, you will see which is the phenotype. When we say the phenotype is what happened in the cell when you don't have the protein of your interest. And as you can see here, this is a control cell. The red dots are the lipid droplets in cells with TANGO2. But when you remove, you can touch one. So this is a 3D reconstruction. You can see cells that are doing this in the microscope. And you can see that every dot is a lipid droplet in a control cell or in a cell that has TANGO2. But when you remove TANGO2, you can see that the lipid droplets are bigger. Here, if you can see here, the dots are bigger than here. So then this was the first result that or that made me think that maybe was TANGO2 related with the lipid metabolism. Next. So, second conclusion was that when we removed TANGO2 from our system, then the lipid droplets were bigger. Next, please. But I told you that, 5 minutes. So, let's go. So, lipid droplets, I told you that it's like a ball. So, it's a ball, but it's a particular ball because lipid droplets are our batteries. We put lipids in, and there are lipids that go out. So how we can change the size of a ball that we can put lipids in and out? We have two possibilities. Or we put more lipids inside, or we block the outside of the lipids. So there are not more possibilities there. So we tried that. Let's go. So to change the size of a lipid droplet, we have just two possibilities, and we test both of them. If we add more lipids, means that we are producing more lipids, so we call that biogenesis of lipids. And if we are not consuming the lipids, we are making like a block in the outside of the lipids. So we make both experiments. Then, if you want in the break, I could explain you better. And the final conclusion here was that when we don't have TANGO2, both situations was altered. So we have an increase in the biogenesis of lipids. We are producing more lipids when we don't have TANGO2, but also we are consuming less lipids when we don't have TANGO2. Next, please. But then I remember that I read that some colleagues published that when you don't have TANGO2, you increase the amount of ROS. And I told you that I will explain you what is ROS. So imagine that you use a car, and when you use a car, you burn fuel, and the byproduct of use a car is the production of CO2. And if we don't manage well the production of CO2, then we will see irreversible damage in our environment. So in the cells happens more or less the same. Mitochondria produce energy, and the byproduct to produce this energy, it's called ROS, reactive oxygen species. If we don't manage the amount of ROS inside a cell, then we will see that there are changes that are damaging lipids and also proteins. So one thing that we did was to reproduce the data of our colleagues, and we realized that was true. When you don't have TANGO2, you have an increase in ROS. But then my question was, okay, but that amount of ROS that is increasing, it's enough to change our lipids. So I did an experiment that is called lipid peroxidation. So you see if the lipids that are in your cell are damaged in an irreversible way. And what we found was that yes, when you don't have TANGO2, you have an increase in ROS, but this increase in ROS is enough to alter the lipids. And now we know that there are many new publications about the function of lipid droplets, because they are not only the batteries of the cell, but we are still thinking like that. So now the lipid droplet has the capacity to absorb the lipids that are modified in the cell, because those lipids are toxic for the cell. So maybe the lipid droplets are bigger because we are not using it to produce ATP, but also because they are trying to avoid the lipotoxicity that you have lipids free in your cytoplasm. I have 1 minute. So, summary, when you have TANGO2, TANGO2 is predominantly in the mitochondria and in close conformation or in close contact with lipid droplets and the ER. And when you don't have TANGO2, the mitochondria looks disgregated, the lipid droplets are bigger, and the lipids that are inside the lipid droplets are modified. So finally, I want to thank all my colleagues in the lab. I want to thank Vivek. I want to thank all our external collaborator, Sima, because she gave me all the cells from patients, and we did a lot of results with patient cells. The sponsors, because I'm here also because they provide the opportunity, but mainly I want to thank you to all the TANGO2 families for serving us as a constant source of inspiration of our work. Thank you very much.